So hello, thank you very much for giving me this slot. So I will be talking about microbiome time series and analysis of this kind of data. So first of all, the motivation behind our study is that gut microbiome is a dynamic milieu, and if we can predict, all right.
Just a second.
So the motivation was that microbiome is a dynamic environment, and if we can understand it, how it changes in time, and if we can predict it, we can influence its behavior. So, for example, we can plan some therapies such as probiotic therapies or fecal microbiome transplant therapies. So what we wanted to do, we wanted to, inspired by econometric and statistical analysis, we wanted to develop some tools to analyze how gut microbiome will change in time so we can understand a bit better. So what we did, we found a very dense, so a sample from more than 1 year almost every day for the data sets of 16S data sets of healthy people. And then we analyzed how the gut microbiome behaves as the whole community, how different bacteria will behave, and also whether there are some clusters of bacteria that have very similar, behave similarly in time. So first of all, we wanted to analyze how the gut community will behave as a whole. So for this, we chose alpha diversity. So we wanted to analyze how the alpha diversity will change in time. So here you can see how the alpha diversity looks in time. So you can see this is extremely variable. So there is very much variation from day to day. However, this red line is the trend. So this is like the mean of this data. So you can see that despite there is lots of variation, this data, when the person is healthy, it's quite stable all over the trend. However, as you can see, in those yellow spots, for example, on subject 3 and subject 4, there are some events happening. So for example, subject 3, it disappeared. He got some food poisoning also, and the subject 4 also got food poisoning. So you can see the alpha diversity is stable, but when there's an event happening, the alpha diversity richness will go down, but after it will come back exactly to the baseline, which was very interesting for us. So what we wanted to see in econometrics, when the process is stationary, it means that it mean, so this red line, and the variance is stable in time. And also this kind of process can be predicted. So what we did, we wanted to further analyze whether gut microbiome can be predicted. And we analyzed autocorrelation. So autocorrelation is dependence on its previous states. And this is the first column here. So we can see that every subject had very high autocorrelation. And not only he had autocorrelation, but also we saw those fluctuations. And this also in econometrics means that there is some repetitive pattern, some fluctuation that's happening on a repetitive manner. So for that, we analyzed it with fast Fourier transform. So we wanted to find some seasonality in our data. And a small disclaimer here, it's not seasonality like spring and summer, but some repetitive patterns, so like increase and decrease of alpha diversity. And actually, we found that all of the subjects had very multiple seasonalities in the data. And this is 16S data, so we're not that much able to go into function, but we believe that it might have something to do with metabolic interaction between bacteria. Also, what we did, we wanted to find out whether we are able to predict how the gut microbiome will change in time only using its previous values. So for that, we use a RIMA model with Fourier seasonality. So we trained the model on previous days of alpha diversity and also previous seasonality. And we see that our model works fine. However, as you can see on the first panel, there is this food poisoning happening. So, and the model does not know that. So it predicts still the same gut microbiome as if it was healthy. However, it is not, and the model does not capture that. Then we went on analyzing different features, so how specific bacteria will behave in time. And for this, we created what we call a feature vector. So every bacteria is described by many properties of a time series. So mean, standard deviance, prevalence, autocorrelation, whether it's stationary, so whether it mean and variance will stay the same in time, whether there is some autocorrelation, whether this bacteria will behave as a noise. So noise process is completely stochastic. So if a bacteria behaves as noise, we cannot predict it. And also we found we wanted to analyze how much importance as it comes to variance the bacteria have. And that was absolutely fascinating for us to see, first of all, that for every bar here is a subject. And we saw that almost more than a half of bacteria in every subject is noise. So basically, those are bacteria that you cannot predict using some predictive models. And we think that it might come, for example, from some technical variants during sequencing, etc. And also what we saw on the last panel is that most of the microbes are stationary bacteria. And also small disclaimer here, noise is stationary by definition, so also noise is included in those bars. However, apart from noise, most of the bacteria that are in gut microbiome of a healthy person is stationary, which will mean we should be able to predict it. And also what we wanted to do, we wanted to define whether there are some regimes, so whether there are some bacteria that behave similarly in time. And for this, we define 6 regimes of the gut microbiome, so 6 different longitudinal behaviors. So this is stable prevalent of bacteria that are almost all the time in the gut microbiome and are stationary. So those bacteria are always there, they are very abundant, and also they are quite resilient to the environment. Then we have unstable and prevalent. So those bacteria are in the gut, but they change and they are not stationary, which means that they are very prone to perturbations. And if something happens, for example, if you get sick, they will not come back to the baseline. Also, we have stable temporal and unstable temporal. So temporal bacteria we define as bacteria that are not always there. So they appear due to some events. And then we have rare and noise bacteria. And here we made the deliberate choice to split them, because rare bacteria are not noise, because white noise is like stochastic process that we cannot predict. But rare bacteria, as I will show you in a second, are bacteria that appear due also to some events and then disappear. So what we wanted to do, we wanted to see how much of all of these regimes we have in the gut microbiome. And it was also quite a surprise for us that almost when you quantify them, most of the bacteria are noise and rare, as you can see from the bar plot, and only the small part are those stable bacteria. However, when you take a look at the abundance, so the plot on the right, this blue area here are stable and prevalent bacteria. So even though they are not numerous, their abundance is exceeding everything else. And also the last 2 bar plots are those, we have some blooms of very rare bacteria. So as you recall, there is some traveling and food poisoning. And for example, around day 100 in the third subject, you can see a sudden bloom of rare bacteria that then disappears. But all over, this composition is quite stable in time. Also, what we wanted to do, we wanted to define some bacteria that fluctuate together in time. So for this, we wanted to analyze which bacteria will cluster together in time. And we can see here that lots of bacteria, so this will be the cloud here, are not correlating with anything. So they are not fluctuating with anything else. And those, as you can imagine, are mostly rare bacteria. And then you have those very small clusters in every subject that is comprised where you have this bacteria of signal. So those are bacteria that are stationary or non-stationary, but they are most of the time in the gut microbiome. So what we think here is that all of the noise, if you want to model, for example, gut microbiome and predict it, you can remove all of the noise, so all of the cloud here, and only focus on the small cluster that we believe can be predicted. So first of all, we created this framework, a statistical framework on stickiness data to analyze gut microbiome, and then we wanted to move to predictions, so just to predict how the gut microbiome will change in time. And for this, we were searching a lot for some data sets of time series data. However, there are not much of them. So we created a workaround. So we wanted to use a large cross-sectional data, then predict how the gut microbiome will change in time. So for that, we switched from 16S to shotgun. And first of all, we took a large cohort of 6,000 people. And then every person is defined by its taxonomy and pathways. And then using an autoencoder, we wanted to create an embedding that will represent every person as its metabolic, so functional and taxonomic potential of the gut microbiome. So it will reduce all of the noise that I just showed you. And then we wanted to use this embedding for downstream tasks such as regression, so age prediction and BMI prediction, classification, healthy versus sick, and then time series prediction, so FMT trajectory prediction. And this is the most important for us because fecal microbiome transplant is a very important therapy that's getting recognition right now. So what you have, what it is, you have a healthy person that is the donor of the fecal sample, and then you will take the sample and you will put it in the patient that is sick, and then this person you hope it will get better. However, normally it's not being done in silico. You just take a healthy person and just prepare the FMT and you do the FMT. However, what we want to do, we want to really understand what is happening during the FMT to maximize the success rate. So for this, in FMT, you have 3 samples. You have donor sample, patient pre-FMT, and patient post-FMT. So what we did here, every sample was embedded during our model, so this model. So every sample is defined as an embedding, which defines the functional and taxonomic potential. And then using a simpler model, we wanted to predict the outcome of the FMT. Also, we were predicting the embedding. And then the embedding was decoded by the autoencoder model, and we are getting the prediction of the FMT, how the patient will react to the FMT. And here you can see our results. So first of all, we wanted to check how our model will perform on an unseen dataset, on the FMT dataset for the reconstructions. And here we can see that for the donor and for the patient post-FMT, so people that should be generally healthy, this model performs better. So here, as a measure, we use procrustes disparity. So this is a measure that will tell you whether the model, the reconstruction of the model, will understand the distances and the differences between people. So you can see that for donor and for post-FMT, the model is working fine because the best score is 0. However, for the person before FMT, the model is struggling a bit. And this is also understandable for us because we see that before FMT, when the patient is very sick, there is complete chaos happening in the samples. And what was the most important for us, the last scatter plot, you can see the predictions, so how the model predicted FMT, the patient after FMT. And here you can see that the model was able to decode, like predict and decode FMT, the patient after FMT. Yes, so as a summary, we created this statistical framework to analyze time series, and we see that the healthy gut microbiome is relatively stable in time. There are some seasonal patterns which are not related to seasons. Most of features which are in the gut microbiome are noise, and only a small subset we believe is signal. And also there are some cluster of bacteria that have similar properties. And also we show that using cross-sectional data, you can use cross-sectional data to then use it on a time series prediction in the future. So I wanted to thank all of my collaborators, and thank you very much for listening to me. And this is the link to the GitHub of the repository when you can download the code for the analysis.
Thank you, Zuzanna, for a nice presentation. Any questions?
Hello, thank you very much for a nice talk. I was wondering how robust the results were that you were getting. So I'm asking about the first longitudinal part. So the stability of the microbiome, seasonality, amount of noise, to the method that you're using to define it.
Well, it's very hard to talk about robustness right now if you have only 4 people. However, our method for now was used on very dense time series, and there are not many more than dense time series. So this is very hard for us, but I know there are lots of new data sets that are coming right now, and we are very eager to use our method on those data sets.
Thank you.
I would be curious to hear about the time series modeling. How are you dealing with missing samples or irregularly sampled time series?
We are using imputation.